All right, hello everyone, and welcome back to Chapter 9, Part 2. And we're also going to cover Part 3 in this lecture as well, just because Part 3 is a rather short section. I am going to warn you, 9-2, this is going to be a pretty long, dense section. There's going to be a lot of really dense information, but it's going to be really helpful. So get your notes ready and let's begin. So we're going to be talking about the visual system's functional anatomy. It's important because vision is really our primary sensory experience for humans, and far more of the human brain is dedicated to vision than any other sense. It's really kind of the pinnacle sense that humans have. The same way that for dogs, they just have these incredible noses, humans have this really incredible vision ability or sight. And understanding the visual systems organization is going to be really important in just understanding the human brain function altogether. It's out of the parts of the brain, there's so much mystery of the brain, but neuroscientists feel fairly confident that we have a pretty good understanding of the visual system and what's going on there in the human brain. So let's go back to basics, way back to literally some of the physics of what's going on in a very basic way. Let's talk about just visible light and the structure of the eye. Now, all around us, there is all kinds of electromagnetic energy. Some of it we're able to see, most of it we can't, right? Light is really part of this electromagnetic energy, and visible light is just a small portion of that. So here we can see all of the different types, or at least many of the different types of electromagnetic energy that's in the world around us. And we see this small little section that includes the visual or visible light that us humans are able to see. Now, notably, this isn't a hard rule for all creatures, as they state here. Butterflies, honeybees, they're able to detect light in the ultraviolet range. And so they have a broader range of light that is visible to them that we aren't able to see. There's also the infrared light that's around us. And with the help of technology, you have heard of those night vision goggles or whatever, and we are able to see some of the infrared light with the assistance of those devices where we might not be able to otherwise. Now, the range of electromagnetic energy that's visible to humans ends up being only about 400 nanometers. That on the low end is going to be the violet light, all the way to 700 nanometers, which is red light. And we can see the whole range in between here if you're curious. And for those of you who are interested or who are trivia buffs, a nanometer is 1 billionth of a meter. So just a little fun fact for you there. Now, let's start to get into some of the neuroanatomy of what's going on with the eye. And I'm going to try my best to break this down, and we'll be going over some of these parts a bit more continuously as we go through this section of the lecture. So let's begin by using this image and some of the terms we have here. So cornea, that's going to be the clear outer covering. We can see that right here on the eye, that clear coating right on the outside. Then next we have the iris. Now the iris, that's the part that we normally think of as being really pretty. That's where a lot of the color is in our eyes. This part is, we can see right here, what opens and closes to allow more or less light in. And of course, where is that letting that light in? It's letting light into the pupil. Our pupils will expand or dilate, and that's going to be depending on the movement of the iris. Now, behind the pupil, that's where our eyes have a lens. The lens is used to focus the light. It's trying to bend to accommodate to near and far objects. And it's important to think about this in a similar way to muscles. So much of our lives in the modern day, too, we're spending a lot of time just kind of looking at screens or in indoor environments. And so that's putting a type of strain on some of our lenses as well. So there is actually some research that's out there to suggest that it's really good for your eyes to make sure that you're spending at least a few minutes each day if you can, trying to get little 5-minute breaks to just be outside. And that helps just to change the type of bending that your lens has to do to help preserve your eyes as you get a little bit older. Then for your retina, we can see that way back here. That's where the light is being projected to, and where light energy initiates some of that neural activity. So this is a really helpful image to just kind of visualize some of the anatomy of the eye and the process of the light. We're going to go into this in a bit more depth here as well. So let's talk about some of the other parts of the neuroanatomy and some of the ways that all of this works together, try to give you some helpful examples to be thinking about as well. So we have the sclera that forms the eyeball. That's the whites of our eyes is what that's referring to. There's also the cornea, which we talked about before. That's that clear outer covering over the eye, and the colored iris. You can see that nice blue here, which opens and closes to allow more or less light through the hole in the center or the pupil. Then we know that behind that is the lens that we had just talked about. Now the lens is bending that light or focusing it onto the retina. In the retina, that's where then this light energy is initiating the neural activity. And at the center of the retina is the fovea. Now the fovea, this is the region of the sharpest vision, and it has the densest distribution of photoreceptors that are specialized for color. All right, so and we'll go over to this for a little bit, but we'll return back to the fovea as we're thinking about that. Here we have a photograph of the retina's surface. We're able to see some of the distribution of the blood vessels that exist on the retina. And we can see here that we do have this blind spot, which is really interesting. That blind spot is where we have this optic disc, and that's where the blood vessels enter the eye, and the axons that form the optic nerve leave the eye. It has no receptors, and so we have a blind area there. And it's very, very small. The reason why you don't notice it most of the day is your brain actually kind of fills in that gap, that little blind space. It kind of uses the other information or the other sensations that are around and just tries to color in something that seems like it would make sense in that space, which is just interesting and cool. Again, if you're interested in that, testing that out, you can Google and look up tests for your blind spot. And I've done some of those before where you're moving pages around in dots, and you're able to get dots into places where you're no longer able to see the dot, and it looks like just an all white screen, for example. Now, in contrast, we do have this fovea where we're able to get this really clear image. And I think of that almost similar to if you have like portrait mode on your phone, right? It's similar with our eyes, where in portrait mode, usually a person, maybe you, are very clear, and the background around you is a bit blurrier. That's similar to our vision. If we really think about it, as we're looking around, we're not able to see the other information that's within our visual field with the same clarity as we are wherever we're directing our eyes. So when you're reading a book, it's not that you can see all of the words on the page as clearly as all of the others. It's really wherever your eyes in directed focus are, where that's taking place, and you have the clearest image. That's where the light is being projected to the fovea. Now, let's go back to the cornea and the lens of the eye. This is very similar to a camera. It's a really great analogy. It's used to then focus the light and to project a backward inverted image onto the light receptive surface, which is also just kind of a fun, interesting thing to know. Here we see the key. So this person is looking at a key, and that light is going through the parts of the eye. Then we can see that it is being inverted and flipped backwards. So now the key is flipped and inverted here as it's projected onto the retina. And as light's entering the eye, it's going to be bent first by the cornea. It's just going to travel onto the pupil, and it'll be bent again by the lens. Now, the cornea itself, that's fixed. That's not going to be moving, but there's going to be all of those small muscles that are going to be adjusting the lens, which is what I was talking about earlier in making sure you're stretching that lens in your eye. Now, what about for those of you who may have glasses? Let's talk about that for a little bit and what's going on there. So let's begin with maybe typical vision. This is when the lens is focusing incoming light directly onto the retina, like we can see back here, and it's working really well. But some people have myopia or some of this nearsightedness. They cannot bring distant objects into clear focus. And that's because this focal part of the light, it's falling short of the retina, as we can see here. Now, this is usually caused by the normally round eyeball being elongated a bit. And this can also be caused by extensive curvature of the front of the cornea. Alternatively, people with hyperopia, they cannot focus on nearby objects because the focal point of the light falls beyond the retina. We can see here where the focal point where it would be is further back. Whereas the myopic eyeball may be too long, the hyperoptic eyeball may be too short. So this farsightedness may also be due to the fact that the lens is too flat to really refract the light adequately. So I think it's just some interesting information there as well. I also want to go back to that optic disc. It is just really important to recognize it's not that our eyeballs are just kind of floating there in our head. It is all connected, and there's this direct line through the optic disc that's causing the blind spot that's bringing all of that information directly into our brain. It's really a highway of visual information that's going through there. So we're very happy to have that. Now, we went over this a decent amount in that last image, so I'm really going to kind of go through these rather quickly. Pause or feel free to rewind if need be. So we have the retina, that's going to be the light sensitive surface that's on the back of the eye. On the retina, there's going to be the neurons and the photoreceptor cells, such as the rods and the cones and whatnot. This is translating the light that exists as physical stimuli into action potentials that are then, like I said, it's translating the language into what the brain speaks to be able to talk to the brain and the nervous system. It also helps, pardon me, to discriminate wavelengths and colors, and it works in a wide range of light intensities. So we're able to see things still when it's dark out and when it's very, very light out to some extent. Now, the fovea, that's the region at the center of the retina that's specialized for high acuity. This is the receptive field at the center of the eye's visual field. So let's talk about that a little bit more. The central focus in the fovea, this is the cross section through the retina. In image A, up above here, this shows the depression at the fovea, which is also shown in the scanning electron micrograph. And that's showing where these photoreceptors, they are packed most densely and where our vision is going to be the clearest. So we can see there's all of these receptors on the fovea, and they are just ready to go to get that information and to get the clearest HD information to our brain. So we're able to get that. And it's all based on where we're putting our attention as well, right? We're thinking about how helpful this is going to be, because if we were to theoretically get all visual information with the same amount of clarity, it would be much harder for us to be able to direct our attention the way that we do right now. It would really change a lot of how we would imagine what that process might look like if all information is as clear as where we are trying to direct our attention at any given time. Now, as we're talking about the acuity across the visual field, vision is better at the center of the visual field than it is at the margins or at the periphery. So in this nice example here, when we look at the center, the letters in the periphery must be much larger than those at the center for us to see them. I talked about reading a book earlier and how you're not able to read the words on the bottom of the page as you're reading across the top line of the page, if your eyes are fixed there. So if you take a moment to focus on the star in the middle of the chart, this will help you to understand the relative sizes of letters that are legible in the central field of vision compared to the peripheral field on the outside, and how these larger letters really help us to be able to see them if they're in the periphery. Go into the structure of the retina a bit more. So there is this blind spot, which includes a region of the retina or the optic disc that we talked about, where axons are forming the optic nerve to leave the eye and where these blood vessels enter and leave. That section, it has no photoreceptors. We're not going to be putting photoreceptors on top of all of these axons and blood vessels and whatnot. It's already in there. There's enough going on. Let's leave that be. And we can put photoreceptors in other places like the fovea. We don't need the HD vision here. And it's a relatively small section of our visual field. Now, there is a condition called papilledema. I did not pronounce that right, but I tried my best. That is when there is a swollen optic disc. This may be due to high intracranial pressure, maybe things like a tumor or a brain infection, or inflammation of the optic nerve, such as optic neuritis. It can also cause loss of vision. So I talked before about finding your blind spot, and it's an interesting kind of exercise. You can find figure 9.4 in your text, and that's going to help you do some of that as well, which is fun. But your visual system, as I had alluded to, solves the blind spot problem by having the optic disc in a different location in each eye. The optic disc is lateral to the fovea in each eye. So it lies to the left of the fovea in the left eye and to the right of the fovea in the right eye. Oh. Now let's get into photoreceptors. With the photoreceptors, we have rods and cones, essentially. Now, rods, there's a lot more sensitive to low levels of light or dim light. So we're mostly using them for night vision, or when there's not much light available for us to be able to use. Then because of that, there's only one type of pigment that exists. So you'll notice this, especially on these nice summer days where we're starting to have an extended period of time with dim light. You'll notice that as you go out, you're not able to see the green of the leaves or the grass quite as well. It's all becoming one kind of color, and you can see lighter and darker, okay, and that contrast can still exist, but you're not able to get quite the same acuity. Whereas with cones, these are highly responsive to some of that bright light. They're specialized for the color. They have the high visual acuity, and these are in the fovea only. They have the three types of pigments, the red, the green, and the blue, that we're able to utilize in humans. So here we have just a nice little figure of these photoreceptive cells that are going on. We see the rods and the cones. Now, rods and cones, they're these tube-like structures. As the scanning electron micrograph shows, they differ, especially on the outer segment containing light-absorbing visual pigment. Rods are especially sensitive to the broad spectrum luminance, whereas cones are more sensitive to the particular wavelengths of light. Now, the way that I think about this, just to keep rods and cones straight, the little tool that I use is I think of a rod as being a little bit simpler. In my mind, if you have a rod, it's just kind of this straight thing. So a little simpler, light, dark for rods. Whereas cones, I think of that as being a little bit more involved, right? There's going to be this circle at the bottom, and it's all coming to a tip at the end. So there's more things going on. There are these multiple colors and the fine detail that is required for that. So I don't know, maybe that just helps me. I hope that helps you. So I thought I'd offer it along, but I apologize if that just is more confusing. We talked about this now a little bit, but let's go over it again. There are the three types of cone pigments. These absorb light over a range of frequencies, but we can also see the maximal absorptions that are here. So it's 419 nanometers for blue, it's a shorter wavelength, 531 for the green. These are the middle wavelengths, and then 559 nanometers for the rather long wavelengths, relatively, on the peak sensitivity of each of those. And notably, there are approximately equal numbers of red and green cones, but fewer bloom cones, interestingly enough. And let's take a moment now to think about this from a clinical angle as well. So the eye works correctly only when sufficient light passes through the lens and is focused on the receptor surface or the retina. Now, too little light entering the eye produces a problem in visual illuminance, in which the objects are too dim and it's hard to see any image at all. Now, with age, less light is allowed to travel through the lens and cornea. And when you think about this, how could this affect people who are driving as they're getting older? We have the contrast of these two images and the clarity of those that are taking place as less light is entering through the eye. It's important to recognize that visual luminance, this is typically a complication of aging eyes, but it's one that corrective lenses alone cannot cure. The eye's lenses and cornea allow less light through as we age. So as a result, there's less light that's available to strike the retina. There was a researcher, Don Klein, who estimated that between ages 20 and 40, people's ability to see in dim light drops by 50%, and over an additional 20 years by a further 50%. As a result, seeing in dim light becomes increasingly difficult, especially at night, and the only way to compensate for visual illuminance is to increase lighting. That's also a reason where I've had conversations with some older adults, and they've talked about when driving, no matter the time of day, whether it's in the morning, afternoon, or evening, when you're driving, if you are driving and you have your headlights on, it's just easier for some of them to catch and see because there is more light that's being there. And for younger folk, they may not recognize how helpful that can be, because for us, there's enough light that's still able to go through and strike the retina, where it doesn't make a large difference. But I've talked to older adults where it does make a big difference. And now some of these cars have the automatic lights in them, but that's just something else to consider about a way you could maybe help people around you as you're going about your day and driving. Retinal receptors. Now, this is the retinal mosaic of rods and the three cone types. We have this diagram here, which represents the distribution near the fovea, where the cones are outnumbered or outnumbering the rods. Red and green cones outnumber the blue ones, and the gene for the red cone is carried on the X chromosome. Now we're thinking about that and thinking about some of the color blindness as well. If the gene for the red cone is carried on the X chromosome, that means that then if your dad is colorblind, then there may be a greater possibility that that gene is not being passed along for you to be able to get the red cones as well. Which is also part of the reason why then for males, there's just a higher probability of color blindness, interestingly enough. Getting into some of the types of retinal neurons. There are bipolar cells. These receive input from photoreceptors. Then, so photoreceptors, rods and cones, right? There are horizontal cells. These link the photoreceptors and the bipolar cells. There is the amacrine cell, which links bipolar cells and ganglion cells. Then there is the retinal ganglion cell, or the RGC. This gives rise to the optic nerve. And it's an interesting image to see here. You might think, oh, is this backwards? No, it's actually not. So there is the light that is taking place, and we're seeing that there's the rods and the cones back here, how they are going. They're connecting to some of the horizontal cells, getting connected to the bipolar cells. And as we go up, we have these macron cells, the ganglion cells, then as well. Now, glaucoma. You may have heard of this. Glaucoma damages the optic nerve and it's the most common cause of irreversible blindness. Treatment approaches and research does exist that's going on right now. It's important to think about with this. So the optic nerve, which conveys the information from the eye to the brain, begins with the axons in the retinal ganglion cells, or the RGCs. So if the RGCs are dead or dysfunctional, then vision is impossible. So the clinical goals with glaucoma is to try to repair or replace the RGCs after injury. The current approaches in research are primarily using some of these mouse models where they're trying to transplant healthy retinal ganglion cells or RGC precursors through a donor that we can see right here, which would then help to stimulate non-human embryonic stem cells to generate RGCs in vivo or in a living person, then activating the mammalian target of rapamycin, signaling the pathway while exposing the eye to high contrast images. An implant prosthetic device into the eye would then introduce light sensitive ion gated channels to repair receptors. And then we would use CRISPR to reprogram genes that are expressed on the rods. So there is research that's going on with glaucoma to be able to cure some of the damage that's caused by it. And some of it's looking really promising. So that's something to be thinking about in the coming years. Let's talk about the different types of ganglion cells. So there are the magnocellular cells, or the M cells. Magno, let's think bigger, large, right? This receives input primarily from the rods, and it's sensitive to light and moving stimuli. In contrast, there are the parvocellular cells, or the P cells. Parvo, small, it receives input primarily from the cones, sensitive to color. Think about this in this way too, right? So if you're watching an older TV, maybe it's a black and white TV, you're getting the idea of what's going on. You're able to see the characters. It's going to be in black and white. You're seeing the characters that are able to move around. That's fine. That's big picture. You're understanding what's going on in the story. Big picture, magnocellular cells. Then there's the parvocellular cells, the P cells, small, a little bit more in depth, a little more detailed, right? That's bringing the image into HD. We've got the color, we've got the contrast. We're able to get that information there and put that together. The optic nerve, it connects then to the lateral geniculate nucleus of the thalamus. And the lateral geniculate nucleus has 6 layers. So there's 2 magnocellular layers, which receive input mainly from the rods, and 4 parvocellular layers, which are mainly from the cones. And we can see that here, where there are those 2 magnocellular layers and the 4 parvocellular layers. Let's get into the visual pathways. So there's the optic chiasm. When we're thinking about neuroanatomy, optic chiasm, that's going to be neuroanatomy 101. We're going to want to make sure we understand this. The chiasm, that is the junction of the optic nerves from each eye. This is where the axons from the nasal or the inside half of each retina cross over to the opposite side of the brain, whereas the axons from the temporal or outer half of each retina remain on the same side of the brain. We're going to be able to see an image of that or a figure in just a moment. The takeaway is going to be right here. The information from the left visual field goes to the right side of the brain. The information from the right visual field goes to the left side of the brain. Notice I'm emphasizing visual field. I'm not saying left eye and right eye. That's not really how it works. So we have this person, let's call him Jimmy. Jimmy's looking out and Jimmy has this visual field that exists, right? So when this is taking place, there is the nasal images, where we're going to have the light that's going to be going by the eye, then there are going to be those more peripheral bits of light that are taking place. And we can see that the light that is taking place here on the inside by the nose, right, nasal, that information is crossing over to the right side of the brain. It's crossing, this is the optic chiasm right here in the center, right? That's where the railways of visual information are crisscrossing. The nasal side, that's crossing over. Same thing here. The nasal side, that is crossing over. Whereas the light information that's coming and hitting the outside of the eye, so in this case, the right visual field, it's on the outside, it's staying on the outside. It's not crossing over. And likewise over here, okay? Now, when we're looking at this type of image, it's really important that we're really thinking about this from almost the physics level. When we're looking directly forward, our eyes are rounded. So the angle in which the light is coming in and it's hitting our eyes, it's going to be on these different angles as well. Not all light that you're seeing is hitting your eye the same way. It's not all being projected onto the retina evenly. Obviously, there's going to be different parts of light that's around us that's going to be projected onto different parts of the retina. So we can see that then this is where the light is really hitting the back of each of these eyes, or Jimmy's eyes here, as it's taking place. So some information is crossing, but it's taking the information from the left visual field, and all the information from the left visual field, that's crossing over to the right side of the brain. Whereas all the information from the right visual field is all crossing over to the left side of the brain. But it's not just stuck with each eye. It's not all the information in the right eye is staying on the right eye or all crossing over or all staying on one side, nothing like that. I think that this is a complicated figure to understand, but it's a really important one to try to spend some time delving into and getting, right? So I'll just repeat it one last time. Information from the right side of the visual field in blue moves from the two halves of the retinas ending into the left hemisphere, whereas the information on the left side of the visual field is hitting the right halves of the retina, right half of the left eye, right half of the right eye. That information is staying on the right half of the brain. Okay. Let's talk about then the three routes to the visual brain. There is the geniculostriate system. These are projections from the retina to the lateral geniculate nucleus to the visual cortex. There is the tectopulvinar system. These are projections from the retina to the superior colliculus, to the pulvinar thalamus, to the parietal and temporal visual areas. Then there's the retinohypothalamic tract. These are synapses in the tiny suprachiasmatic nucleus and the hypothalamus, and the roles in regulating the circadian rhythms and in the pupillary reflex. And we can see some of these systems that are then shown here. So the optic nerve follows the geniculostriate path to the primary visual cortex and the tectopulvinar path to the temporal and parietal lobes. The striate cortex, this is the primary visual cortex. It includes two visual paths which emerge from the striate cortex. There is one route that goes to vision-related region of the parietal lobe. One route goes to the vision-related regions of the temporal lobe. There's also these dorsal and ventral visual streams. The dorsal visual stream is the pathway that originates in the occipital cortex and projects to the parietal cortex, right? Dorsal along the top, parietal lobe up here. This is the how pathway, how an action is being guided toward an object. Then there is the ventral visual stream. We can see here, this is the pathway that originates in the occipital cortex and projects to the temporal cortex. This is the what pathway. It is what is identifying an object. And yeah, we see a very similar thing for both monkeys and for humans in these streams which exist. With the geniculostriate pathway, there's the lateral geniculate nucleus or the thalamus. The right LGN includes input from the right half of each retina, whereas the left LGN receives input from the left half of each retina. And there are these 6 layers that you can see more about here. With this, the P and M retinal ganglion cells, they send separate pathways to the thalamus. So the segregation continues in the striate cortex. And the right and left eyes also send separate pathways to the thalamus. And the pathways, they remain segregated in the striate cortex. And we can see here, this is similar to what I talked about in the diagram, where the information travels from the right side of each retina to the right lateral geniculate nucleus. And along that way. And then the information goes from the contralateral side, or the information from the contralateral side goes to layers 1, 4, and 6, whereas information from the ipsilateral side goes to layers 2, 3, and 5 in the lateral geniculate nucleus. Layers 1 and 2, these are receiving input from the magnocellular pathway. Remember those bigger pathways? That's going to be more about the light and movement, while layers 3 through 6, they receive input from the parvocellular pathway, bringing it into the HD, the color, right? The tectopulvinar pathway. The magnocellular cells from the retina project to the superior colliculus, which sends information to the pulvinar region of the thalamus. There's the medial pulvinar, which sends connections to the parietal lobe, and the lateral pulvinar, which sends connections to the temporal lobe. In the occipital cortex, this is composed of at least 6 visual regions. There's going to be the primary visual cortex, which we abbreviate or may call V1. This is the striate cortex, which receives the input from the lateral geniculate nucleus that we just went over. There's also the secondary visual cortex, V2 through V5. These are the extrastriate cortex, another name for it. These are the visual cortical areas outside of the striate cortex. And there's the extrastriate or secondary visual cortex. These are the remaining occipital visual areas. And each region processes specific features of the visual information. And here we can see a breakdown of some of those visual areas, both lateral view from the outside of the cell, and then a medial view if we were to split the brain open of those different visual areas. We can see how the information is really spreading out. It's all arriving at the V1, going from there. Now, when we talk about the heterogeneous layering, there is the blob of V1. It's the region in the visual cortex that contains the color-sensitive neurons. It's revealed by staining for cytochrome oxidase. And there's the interblob of V1. This is the region that separates blobs, participates in the perception of form and motion. And these are illustrated in the drawing to the right of a flattened section through the visual cortex of a monkey. There is that special stain that reveals the blobs and stripes for cytochrome oxidase, which is a marker for mitochondria, the organelles in the cells that gather, store, and release energy. And there's the segregation of information about color, form, and motion, which is maintained at V1 and V2. Here's another nice chart of the visual streams that take place. Remember I talked about that dorsal stream and the ventral stream. Dorsal stream is the one that controls visual action. It goes along the top and it begins in region V1, flows through V2 to the other occipital areas, and then finally to the parietal cortex ending in area PG. The ventral stream, which controls object recognition, goes along the bottom here, begins in region V1, flows through V2 to the other occipital regions, and finally to the temporal cortex ending in area TE. The information from the blobs and inner blobs in V1 flows to the thick, thin, and pale zones of V2, and then to regions V3 and V4 to form the ventral stream. Information in the thick and pale zones go to region V3A and V5 to form the dorsal stream. Damage to these regions, they can produce surprisingly specific deficits. So an example of this is damage to the fusiform face area, which is sometimes abbreviated as FFA. It leads to something called facial agnosia, which another term for it is called prosopagnosia. And that's a condition where an individual cannot recognize faces. So in those cases, they'll sometimes use other cues about a person, even though they don't recognize their face. Maybe they like to wear a specific hat and they are able to notice the hat and realize, oh, yeah, that's Jimmy. Some of the vision beyond the occipital cortex. So in image A here in the temporal lobe, that fusiform face area, or FFA, processes faces, and the parahippocampal place area processes seams. So we can see the FFA in red, the PPA in green. In image B, in the parietal lobe, the lateral intraparietal area contributes to eye movements. The anterior intraparietal area is involved in the visual control of grasping, and the parietal reach region participates in visual guided reaching. And that's the end of 9.2. Let's briefly go through 9.3 because 9.3 is not a particularly long section. So this is talking about the location in the visual world. And we are going about those visual fields. We had that in the other image with Jimmy, where there was the left and the right visual fields. And these are the regions of the visual world that are seen by the eyes that are divided into left and right. Information in the left visual field, that goes to the right hemisphere. Information in the right visual field goes to the left hemisphere. You can take a moment now where, as you focus at the cross at the center of this image, the information on the left of this focal point forms the left visual area for you, and that's then traveling to your right hemisphere, while the information to the right of that center point forms the right visual field, the part that's in blue, and that's traveling then to your left hemisphere. The visual field can be split horizontally, and the information above the focal point is in the upper visual field, below the focal point is in the lower visual field. Each of the retinal ganglion cells, or RGCs, respond to the stimulation on just a small circular patch of the retina, the cell's receptive field. And then there's this coding location. So light falling into one place of the retina will then activate one ganglion cell, while light falling in another place will activate a different ganglion cell. That's really helpful, as the cells in the lateral geniculate nucleus also have these visual fields, and each LGN cell represents a particular place. So it projects to V1, forming a topographic map. The receptive fields of the cells in the cortex are typically larger than those of the retinal ganglion cells, and more cortical tissue is devoted to cells in the fovea and then in the periphery, which we had talked about before too. There's also this topographic organization of region V1, where the fovea sends information to a disproportionately large part of the occipital cortex, which is why the visual acuity that we have is really best at the central part of the visual field. And we can see some of that here, where there's this fovea, and this section a little bit clearer. That's going directly to the V1. whereas this peripheral information is being sent more to the periphery more broadly. It's not quite as strong and there's less space dedicated to it. The cells in the cortex have much larger receptive fields than the retinal ganglion cells. The receptive field of a cortical neuron must be composed of the receptive fields of many RGCs. Researcher Harry Jarrison, he proposed the principle of proper mass, which states that the amount of neural tissue responsible for a particular function is proportional to the amount of neural processing required. The more complex a function is, the larger a specific region performing that function must be. So the receptive field of many retinal ganglion cells combine to form the receptive field of a singular lateral geniculate nucleus cell, and the receptive field of many LGN cells combine to form the receptive. Then we'll go to the colossal connections, the corpus callosum. The corpus callosum, and we're talking about, again, the neuroanatomy 101. The corpus callosum is one of those you are going to want to make sure you know. That is the superhighway of the brain, right? The corpus callosum connects the two hemispheres, but only specific brain structures. Most of the frontal lobes are connected. The occipital lobes have almost no callosal connections, with the exception that cells that lie along the midline of the visual field are connected via the callosum, so that their receptive fields overlap. In here we can see the corpus callosum in the human brain, and in B we can see the darker areas that are shown in the rhesus monkey cortex regions that receive the projections from the opposite hemisphere through the corpus callosum on a similar pattern of projections that connect to the two hemispheres of the human brain. So that is 9, 2, and 3. Thank you for sticking with me through that. I know there was a lot of information. And stick around again for 9, 4 coming up now.